She joined the faculty at UT in 2012 and has been making waves and news ever since. The College of Arts and Sciences honored Dr. Lloyd in 2017 with an Academic Outreach Teaching Award for her efforts, which cover a broad range of activities. In her lab, she and her students focus on research that integrates geomicrobiology, molecular biology, and geochemistry to determine how microorganisms influence marine geochemical cycles. Her work landed 2 spots in the 2018 UT News Most Covered list. In September of 2018, a study published in M Systems concluded that uncultured microbes dominate nearly all the environments on Earth except for the human body. The study is the first time the population of uncultured microbes has been estimated. Newsweek carried the story of, quote, microbial dark matter, I know you hate that term.
I actually like it. Okay. My colleagues hate it.
To its estimated 6 million readers. In December of the same year, Lloyd's work with the Deep Carbon Observatory on deep sea microbiology was highlighted as the group announced a staggering finding. Microorganisms living underneath the surface of the Earth have a total carbon mass hundreds of times more than that of humans. The research was discussed in The Guardian, IFL Science, Mother Jones, Cosmos, and Astronomy magazine. In a more recent study published in Nature this week, actually, Dr. Lloyd and her team discovered microbes can impact major geological processes happening on Earth. Oh, and she's also presented 2 TED Talks. So today, it is my honor to welcome Dr. Karen Lloyd to the UT Science Forum to present Life 1 Mile Below the Surface of the Earth.
Thanks. I appreciate it. Is this a lasery thing? Oh, that's fine. Okay, yeah. All right. Actually, I can laser like that. Oh, plug that in, and then I can. Yeah, I see. And then I can rotate. Okay. So it may seem like we're all standing on solid Earth right now, but we're not. The rocks and the dirt underneath us are crisscrossed by tiny little fractures and empty spaces. And these empty spaces are filled with astronomical quantities of microbes. Yeah, I'm just going to press it. Oh, right, pointed at that now. Oh, it does work. Such as these ones, which are some microscope pictures that some of my colleagues have taken of various places. The deepest that we've found microbes so far, living microbes thus far, into the Earth is 5 kilometers down. So like, imagine if you like pointed yourself at the ground and like took off running into the Earth. You could run an entire 5K race and microbes would line your whole pathway. I think it's hard to conceive of that kind of distance sometimes. But you may not be familiar with these deep subsurface microbes. You may be more familiar with the microbes living in your guts. So if you take all the gut microbiomes of all the people and the animals on the planet and add them up, they collectively weigh about 100,000 tons, which is a huge, huge mass. But it pales in comparison to the total number of microbes that are present on the surface of the Earth. So if you compare, how do I get it to go? Oh, I just press it harder. These are 2 little boxes of sizes relative to the size of those different categories. The human microbiome is a tiny little dot compared to all the microbes that cover our rivers and our streams and our soils and our oceans. But if you compare this to the amount of biomass that we infer is present in the deep Earth, it's even less. So it turns out that microbially, most of life on Earth is actually beneath our feet. It's down there, down deep. So this is basically the impetus of why I study this stuff. It's just a huge biosphere, and we discovered it, not me personally, but my field only discovered it a few decades ago. So this is just like a completely unknown, uncharted territory. And if you can tell, like, I get as excited about looking into the Earth as a lot of people do for looking outside Earth too. So that's sort of how one can conceive of this stuff. Okay, so my pursuit of studying this stuff deep inside the Earth has taken me to some pretty fun places. So this is me a month and a half ago. This is very recent. Running in the high Andes. There's 15,000 feet, which is higher than any point in the contiguous states. And I could do that for just a few steps before I had to like bend over and breathe for a little bit. But the reason why I was there is because I was with a bunch of geologists studying, we were working in the high Andes. And this is where we were. I should have to press it really hard. So the location of my run was right here in western Argentina, northwestern Argentina, almost right on the border with Chile. And the reason we were there is that there's a string of volcanoes running up and down the Andes. And these are basically places, like if you want to study life in the subsurface, then it makes sense to go where life is going to come up to you and you don't have to go down to it. So these volcanoes are really good at bringing things up from the deep. So that's basically why I'm hanging out with a bunch of geologists looking at volcanoes, even though I'm a microbiologist. So this is one of the sites that we studied, and I'm not going to show you results from this because we just went. So right after we do field work, we have no idea what we caught. I hope it's publishable, but you never know. But this is one of these hot springs that just was fabulously beautiful, looking over this salt area. And this is a wider view of it right here. That's me sitting like a Buddha. You can see the only green in the landscape is coming out of the hot spring because this is the only thing that makes water around. And this is the rest of our team sort of sampling this hot spring and looking. What we're looking for are microbes and gases that are buried deep within Earth and they get moved up and they're bubbling upwards. This is Agostina Ciotti, who's leading the group. She's the Argentinian scientist in charge of this stuff. And so that's what it looks like when we sample these. We just like go up to these hot springs and capture she's capturing the gases right there. And then we also, we take all these samples during the day, and then we have to go back to our hotel rooms and look like a bunch of crazy people with our Ziploc bags and things that look like trash, but are actually very highly skilled, high-level scientific equipment. This is my newest PhD student, TJ Rogers, and this is my former PhD student who ended up doing a postdoc with another member of the team, so then she ended up coming to the field with us. And this is one of my collaborators from Costa Rica, who's also involved in the Argentinian group. But it's just lovely, and you get to see some really great places. Like this is in the high Andes. This is where we were in the Antofagasta de la Sierra in the Puna region. And can you tell what kind of birds these are? Flamingos. I don't know if y'all knew, but there are like normal looking pink flamingos in the high Andes, and there's no vegetation. It is the weirdest thing to be looking at pink Florida-like flamingos, and there's no palm trees or even grass. It's very, very strange, but it's their natural place. It's where they are normally. So this is all a bit weird for me because really I'm an oceanographer. So this is my natural habitat. Getting on ships like the Atlantis, diving in submarines, and I did some work on a ship in Aarhus Bay, Denmark, which is where I did my postdoc in Denmark. And I'm so committed to oceanography. That's a baby. That's 6 months into my first child, I think. It was a short cruise. But it may seem weird to do, to be an oceanographer who's up studying volcanoes, and none of that sort of may make sense. But if you look at a map of the world, and think about where the subsurface sites are, sometimes they have water over top of them and you call it oceanography, and sometimes they don't, and then you call it earth sciences. So I don't really care whether there's water on top or not. I'm interested in the deep bathymetry. And I like pointing out this map, because this map was actually published in 1977, which is before a lot of things happened. Like before we got good satellites, before we got good sonar. This is done with sonar, but it's with very crude, rudimentary sonar, but it is still mostly accurate today. And this was done by a woman named Marie Tharp, who painstakingly, even as late as 1977, the men who were in charge of all the vessels at the time, it no longer is the case that men are in charge of everything, but at the time they felt that women shouldn't go on the ships. So because she didn't go on any of the ships, she painstakingly put together all the data, and then she's the one who made the pretty map out of it, but she never went on a ship. We get to have more fun now. So this is a modern map made with modern methods, and the relief is not as great, but basically she got all the Mid-Eastern ridges and the major features correct. But I put a dot on for every place where myself or my colleagues has made a deep subsurface microbiological investigation. So this is everything we know right now. And it may seem like we're getting pretty good global coverage, but if you think about the fact that these are the only places that we've ever studied, it doesn't look as good. Like if we were all in an alien spaceship trying to reconstruct a map of the world from just these sites, we wouldn't be able to. So this is it. We're inferring what's happening inside Earth from this, and that's it. It's not enough. So how we do this is that we normally will grow these organisms in a petri dish. This is what microbiologists do. This is the normal stuff. But we have a little problem with normal microbiology, and that's best illustrated by pointing out that the total weight in carbon of the entire Earth is 7.5 times 10 to the 22 grams. The total carbon mass of all the microbes on the Earth is 5.5 times 10 to the 17 grams. The doubling time, the growth rate of our slowest laboratory cultures, not E. coli. Oh, E. coli grows much faster, but this is the slow guys, is 3 months, which is hard to work with. If these microbes on Earth all had even the slowest doubling time of culture, they would double the mass of the entire Earth in just 4 years. So we have this problem that most of the things on Earth are not growing. Most microbes are just kind of sitting in this stasis. So what we do when we do this normal microbiology stuff where we ask them to grow in a petri dish is we say, like, okay, you who have been not growing for possibly millennia, I'm going to give you some agar and let's see if you can speed it up. So this is the illustration of what happens when we ask the microbes to do this. So these are the results from the first really directed oceanographic voyage looking at deep subsurface life that was off the coast of Peru in 2002. And I'll show you the data. I won't show you the names of the microbes because that's in the weeds, but I'll make a little box for every microbe that we got. And if the box is bright blue, then that means it's similar to something we already knew before. And if it's white, then it's something totally new and zany. It's microbial dark matter stuff. When we look with DNA sequences alone, and I did contribute to this work, we get lots of different boxes, and a lot of them are really, really novel. There's something totally new, something unknown. So since this was the first cruise, the first expedition to this type of place, we all thought, jackpot. Now all we have to do is start growing this stuff because everything there is something really strange. So let's just start growing them, and then we'll be able to do microbiology. The people who did this, who tried to grow these things on petri dishes, I'll show their results. And every time they got one of these, I'll put the box underneath it. And any new ones, I'll stack up on the end. And then the same thing, if they're bright blue, that means they're pretty normal looking. And if they're white, that means there's something really new. OK, so here's the results of the first culturing attempt to get all these new things. 0 overlap, none. They didn't get anything that we saw with the DNA. Furthermore, everything is bright blue. They just got a bunch of stuff that we already got before. And I'm not maligning the scientists who did this work. It's not because they're not good. It's just that they're asking the microbes to do something that they're not geared to do. And then they ended up getting a bunch of weeds popping up. And then another group did this work as well. And I'll put their results up there, maybe. And they got nothing overlapping with what we get with DNA as well. And so because of this inability to get these microbes that seem to be dominant out in nature in the subsurface environments to actually grow in lab, we are using this word, some of us use this word, microbial dark matter, which I like this word because it speaks to the fact that this stuff is everywhere. It's covering the whole Earth, and it's so unknown, we just don't know what it is. But I don't know. I have some colleagues who feel that it's imprecise. Which is true, but all language is imprecise to some extent. I think that it drives home the point that this is a really big problem in microbiology, and it's really, really unknown. So what do we do? What do we do if we can't advance our slides? I'm just going to do this. So what do we do if we can't do this stuff, this normal stuff? Well, I'm sorry, this is lifted from my TED Talk, but I think it's an efficient way to explain it, so I'm just going to do it. I made the slides for my TED Talks, it's fine. So what we do is we line up DNA sequences with each other. So like, I made these data up. These are fake data. If you had a bunch of species that were completely unrelated to each other, none of these A's, G's, C's, and T's line up. It's totally unconnected. So now if I switch it to the real data, like actual real data, everything lines up really nicely. This is the same stretch of a gene that's present in all these species. And you can tell everybody's got a C here, everybody's got a T here. They're related. They had a common mama and daddy at some point way back when, and it's beautiful, and they're all connected.
Can I do that for you?
Yeah, I think. Advance it, please. And so this is what they are. So the top one is us, and the second one is chimpanzees, which I chose a stretch of DNA. We don't have identical across our whole genomes, but it's easy to find a stretch of DNA where we are absolutely identical to chimpanzees because, you know, we just really closely related. But pine trees, you maybe don't think about as your brethren. You should. Or Giardia, which is a protist. It's a single-celled eukaryote that causes disease, gastrointestinal disease. Or E. coli, which is also another disease-forming organism, Clostridium difficile and Dehalcocoides ethanogenes. But basically, the point is that you can take these DNA sequences from everything, and at some point, they're all related. So that you can take how similar they are and how different they are and make a phylogenetic tree and figure out who's really novel and who's really different. Yeah. So one of the techniques that we use to get this DNA out, I think is really cool, and I can say that unabashedly because I did not come up with this, so I can just fangirl on this method. It is a hard thing to take an environment like this, a mixed population. If you scoop up a little bit of soil, it's got literally a million different types of microbes in it. You've got to take that and chemically extract the DNA from it and figure out who belongs to what, and that's just hard to do. So one thing that we can do is we can take, or I can try, so many options. I feel like this should work. We can put it in this machine called a flow cytometer, which can physically separate out just one single cell from this matrix, which is amazing to me. And then we shine a laser light at it just to see what sort of shape it is. We look at the scatter of that laser light, and that lets us know whether we've gotten a cell or not in our little droplets that are moving past the laser. Is it gonna go? I wonder what the problem is. Maybe I hold it down longer. Then when we get that one cell on its own, we break it open to get its genome out, and then we make a ton of copies of it, and then we cut it up into little pieces and then sequence them. I put an asterisk by this because DNA sequencing technology is getting better and better, and hopefully we won't have to cut it up. We could just sequence it directly. I think that's coming. It's sort of already there, but it's not quite good enough for us to rely on completely. And then we put all these sequences on a computer. Oh, this one died. So there's some strange new types of microbes. We know from the DNA that there are some strange new types of microbes in the subsurface.
Look at there.
Yeah, I know. It's like 50-50, right? But the question is, are they the dominant ones? Like, I say they're dominant because that's usually what we only find, but like, how do we quantify them? Like, we need to like get a quantification of these things, and it's hard to do. So we did this recently, and this is the one that got picked up by Newsweek, which I'm kind of proud of Newsweek for paying attention to these things. So I'll tell you our results like this. So as an easier way to show you the results. So I'll put them into categories of like cultured microbes. Like that would be like somebody we know already, where the DNA sequences match up very nicely together. They have very little differences. So to illustrate this, I'll pick somebody we all know. Beyonce. Do we all know Beyonce? There might be people who don't know Beyonce. No, everyone's nodding. Okay. I just sort of take her as like somebody we all know. So this is like a known quantity. Like, yep, cool, she lives in New York, she sings pop music, we know about Beyonce. Second category would be novel genera to families. So these are like these taxonomic hierarchies. This would be like someone we haven't met yet. Like there's a couple of differences in the DNA, they're kind of new, but they're still a person, we kind of know about them. So for that, I'll illustrate it with a baby. Sort of like, yeah, you know it breathes oxygen, it's probably gonna like bananas, but you don't know if it's gonna grow up to be a pop icon or not. Okay, so the third category would be like novel phyla. So a phylum, like our phylum is vertebrata. So we are, just think we share a phylum with snakes and frogs. So this would be like something where nothing in its entire phylum has ever been in culture. So it's got lots of differences in its DNA relative to things that we know. And for that, I'll illustrate it with an alien, because you don't know anything about it, nothing. You just know it's alive. So to do this, we just downloaded every sequence of this one particular gene. We have a gene called 16S that we use like a name tag. And we just downloaded all of them, all of the sequences. It's great. Like, I don't have to say like, oh, we used this study and that study, but computationally, we can do this. So we just took them all on our local computers. This is from all researchers all around the world. Like when somebody in Japan makes sequences some DNA, they upload it to the same databases that we use. So we can just pull it all down. Okay, but then we had to take each one of those sequences, and there were millions of them, and do a computationally intensive algorithm to figure out how closely related, like whether it was a Beyonce, a baby, or an alien. So to do that, like for each and every individual gene, that was computationally very difficult. And I actually couldn't do this for a while, and I was really bending over backwards trying to figure out, like, how do you test this hypothesis? And then Tony Mezzacappa from the Joint Institute for Computational Science at Oak Ridge gave a talk, and he challenged us as scientists to come up with our most computationally expensive thing that we could do. And I was like, I've got a thing. So they let us use the big instruments. So we got to, I put it next to Marie Tharp just to like, because this is the size of the room. Somebody pointed out when I put this on slides, it looks like it might be the size of like a USB drive or something. This is a room. So like a person standing by it would be, it's taller than you are. We did not use Titan for our work, but we used Beacon and Darter. So that was great. They took this problem, which was completely intractable on my desktop or even in my workstation, which is a pretty souped up computer. I couldn't have done it, and it literally took 2 minutes. It was like so anticlimactic. I was just like, man, I was happy. But so the results for the deep subsurface, like what we found, I'll show them in the form of a pie chart, showing you the different amounts, relative amounts of these different levels of novelty. We learned that, in fact, we were right. The biggest population were the aliens, these new phyla. These were the things that were the most dominant types of microbes in the subsurface. There's known things too. There's the Beyoncés and the slightly unknown things, the babies. And we knew this because when people culture things, they do get these things that we've already known. So, it's not contamination. They're finding real stuff. But this means that there really is a lot of unknown stuff out there. It's relevant. So what are they actually doing down there? This is sort of the next question. All right, you've quantified them. You convinced me. Like, what are they doing? What's the answer? Well, one thing we know is that they're slow, as I've said before. And in fact, my colleagues have calculated their turnover times, their total cellular turnover times. So these are cultures that grow really quickly, like you grow up a culture overnight. But the deep subsurface sediments tend to have turnover times of between a year or sometimes up to 30. It's usually like 30 or 40 years. That's just turnover time. They're not necessarily even dividing. So they're just kind of in stasis. Oh, thank you. Turnover time just means the time to make a new version of yourself. So microbes grow by dividing, generally speaking. So that would be a turnover where they create a whole new cell. But they don't have to divide to turn over. They can just replace all their broken parts over time. So that would be the total time to switch out all the molecules inside the cell. But that's surface sediments. They're already pretty slow. Microbes that are present in really deep sediments can have turnover times of hundreds of years. So like, it's hard because when you say biology and when you say microbes, we think fast, growing fast, like dividing, slime, all these things. But those assumptions don't apply to these populations. They're really, really weird. They're really, really slow. And one reason why, the reason that they're really slow is that their lives are crap. They don't have much food. As you get buried down in sediment, the best things to breathe, oxygen is the best thing to breathe, and that goes away really quickly in most places. And the next thing to breathe is nitrate. That gets used really quickly too. Then you can breathe iron, you can breathe manganese, those get used up. Sulfate, that's a pretty terrible thing to breathe, but it works, you can breathe it. When that's all gone, you have to breathe carbon dioxide, which is a very low energy thing. And you combine this with the fact that there's decreasing organic matter quality. So the quality of the food gets worse and worse, just simply because you don't get new food deliveries. So it's, like eating the leftovers of a picnic that happened thousands of years ago. So what this adds up to is that they don't have much to live on. And we can see this because if we look at the total number of cells with depth, this is the cell concentration with depth, it's in lots of different oceans of the world. It always goes down. It's just decreasing over time, which led my student in his first year of his PhD to say, it's not a biome, Karen, it's a diome. That's great. I love it. They're just kind of dying, but over a very, very long time. And aren't we all just kind of dying? So it's what? Seeking enlightenment. That's right. They're looking for Buddha down there. So he did, so I'll share with you some cool stuff that just got published last week from him in MBio, not the Nature paper. I feel like that's getting enough attention, so I'm going to talk about my other student's paper because it's great. So he did this project in the Baltic Sea. That's Jordan. He's now graduated and moved on to his postdoc. But he took these sites from this drill ship, the great ship Manisha, that were all around Denmark and in Norway and Sweden. And they got really nice cores, like 86 meters down. So this is like 86 meters, like almost an entire football field underneath the seafloor is where they got these beautiful samples from, which is not easy to do. And so what he wanted to know is how does this happen? How can bacteria and archaea persist without growing for 40,000 years in these sediments? And I'll show you some data that I'm not really going to fully explain it, but basically we also looked at the geochemistry of these cores just to see if there's anything that's alive in there. And so on the y-axis is depth. So like up at the top here, this is where the water is in the ocean, and then this would be where the mud starts. And you can see that there's things like ammonium being made, and there's a little bit of methane. So these are indicators that they're alive, like there's stuff down there. This is a good place to go hunting for microbes, even though they are tens of thousands of years old. They're doing stuff. That's how we know it. Oh, so you remember how I said that we can do this thing where we pull out an individual cell and get its genome? Well, he did that. Jordan did that for 4 samples. We would have done it for every single depth, but it cost thousands of dollars to do this procedure, so we only got to do it 4 times. And what he caught, so when he looked at these cells that he managed to catch with this cool method, he caught these really novel microbes. So this is just showing, this is actually the backbone here is like a family tree showing how related they are to each other. But these names, I'm showing you the actual names of these groups because I'm assuming you guys are probably, I mean, you're probably not in my field of research because I'd probably already know you. So these names are meaningless to you, like Y, Aerophobetes, and Metasonantes. I will tell you they're just as meaningless to people like me. These are just placeholders. We don't know, these are uncultured. They have taken on meaning because we'll see these things in different places. But I don't know, it's a whole new group of microbes. So we just use these placeholder names for now. But that's mostly what we caught is these new phyla. And we did get some that have some, well, we didn't get Clostridialis, but Chloroflexi have some cultures, but these would be like novel, not quite at the phylum level. And I'll show you two, well, let me try it like this. We got multiple genomes from each of these major groups, and this is the completeness scale, like just how good the genomes were. Some of our genomes were pretty crappy, but some of them were pretty good. We got like 70%. So we just sort of grouped them all together to study the genomes from these guys. Okay, so that's cool. We got genomes from these really novel groups, but actually we want to know what they're doing. Genes are just like the possibility for what's going to happen. They just are like a blueprint. If you walk around with a bunch of blueprints, you don't say, oh, there's a housing complex over there. Like, no, you've got blueprints. No, maybe nobody built those houses. So to do that, to question whether things were actually getting made in the cell, I teamed up with some colleagues from Texas A&M, Corpus Christi, and the University of Southern California, Laura Zinke and Brandy Reese. And they made metatranscriptomes. So that was where the red arrows are, or the places where they grabbed those. These are basically looking at all the sequences of the RNA. So a genome is like a blueprint. I had a better analogy for this, and I've forgotten it now. I'll just use blueprints. This would be like, oh, yeah. So it's like trying to figure out what's happening in a restaurant. Or try to figure out what people are actually eating in a restaurant. And somebody hands you the menu. Well, the menu's got a bunch of stuff on it, one of which is spaghetti. But you can't conclude just because the menu is there that they're definitely eating spaghetti today because maybe nobody ordered it. But transcripts are kind of like if somebody gives you the menu and then they also give you the slips from all the waiters and waitresses. And you can say, oh, they're ordering spaghetti. They're probably eating spaghetti. But you still don't know if it's quite happening or not because you haven't seen the spaghetti. But you've got a good indicator. So that's what transcripts are, basically. They tell you that a gene has gotten transcribed to RNA. Are we going to die?
Not today.
It's a door slammer.
That's a door, okay. So if you add up the total transcripts in all these different groups, we saw quite a bit. So this answered our first question of like, are they actually doing stuff? And turns out they are, which was pretty great. But then we want to know that, we want to go a step farther. We don't want to just say like, yeah, they've been ordering spaghetti. We want to see the spaghetti. So what are they doing? In cultures, you can do this stuff called metabolomics, which is amazingly powerful. Metabolites are small molecules that are present as intermediates in metabolic pathways. So these are like, if you're breathing, then you've got oxaloacetate and citrate and isocitrate and all these different parts of your respiration pathway. These are molecules that we have in our cells because we're breathing. And so if you assay our cells, you know that we're doing it. And so if you do this with a culture, you get something that looks like a very confused subway map, showing you all these different metabolisms, and some of them are bright red, meaning that those are turned on, and some are like getting smaller, meaning that those are turned off, and you get all this really, really high level information. But that's cultures. I've literally got mud from a few tens of meters beneath the seafloor. You can't assume everything you get is from the same organism, or even if it was inside cells. Like, it's just natural and weird and messy and stuff. And so I never thought I could use this method because it would just be too hard to pick apart anything. However, I don't know if you guys know this lab here at UT, but Sean Campagna's lab in the chemistry department does this. And they collaborate with a ton of people on campus. And one person in particular, Hector Castro, who's a technician in that lab, whenever he would see me, he'd say, Karen, give me some of those deep sea samples. We're going to stick them in our machine and see the metabolites. And I'm like, Hector, that's insane. We can't do that. We'll never be able to say anything. And he's like, finally, he convinced me. And I was like, all right, fine. And so Eric Tague is the one who actually did the work. He's the PhD student working on this. And I sent him tons of samples. This is a bit not as expensive an analysis, so we got to do lots and lots of samples. And they put it in their Orbitrap and their mass spectrometer. And I said, Hector, what did we get? Did it work? And he's like, nope, didn't work. I was like, well, yeah, I mean, I told you it wasn't gonna work. And I was like, did you get anything? He's like, we only got 20 metabolites. 20 is like so many. Like, it's great. So I was actually overjoyed. Normally they get the subway maps, but I'll take 20. 20 is good. So from these 20, I will not tell you about each and every one of them, but I will highlight some that ended up being interesting. So the first one that really jumped out at us was this molecule called allantoin, which I had literally never heard of. This is the first time I'd heard about it, was coming out of our metabolite data. And it was really, really abundant. I know that this is not the easiest way to show this, but what this is a heat map. So the redder these little boxes are, the more of this molecule that we got. And then this is one core, and these are depths. So this is the surface, and this is way deep old stuff. And then this is the surface, and this is way deep old stuff. So we got two forms of allantoin. We also got allantoate as well. And it was everywhere. It was in every single sample, and there was a lot of it. So like, this is potentially really interesting. What it is a breakdown from nucleic acids. So when cells die in the subsurface, they leave their DNA behind, and the DNA will sometimes dimerize to uridines will dimerize to become allantoin. So there's actually a pathway worked out for organisms who eat this stuff. And so now we got to say, all right, we've got genomes and we've got transcripts. Let's find out if anybody in our community is actually eating this stuff. Could this be a food source that we never thought about before? And yeah, it turns out we found these Atrobacteria, which is one of these meaningless names of a novel phyla, these alien groups. And I circled one because these are all the different enzymes that could have to do with metabolizing allantoin, but one is the most crucial one that really says it has to be allantoin. It was expressed in transcripts at many different depths by these Atrobacteria, and nobody else was making it. So what this looked like to us was that only the Atrobacteria can metabolize and get energy from this substrate allantoin. So that means that these are potentially sort of the winners of the subsurface. So we wanted to know what else can they do? What else are they eating? How are they making a living in this god-awful place? And for that, I collaborated with my husband, Drew Steen, who's in the Earth and Planetary Sciences department, and his student, Jenna Schmidt. And they developed a bunch of enzyme assays to look at the rate at which the food was being taken up in the sediments and to see if we could match that up to what we're seeing in the genomes. And they did this at a bunch of different depths for one of the cores as well. And what they found was that when they put in substrates like sugars, so these are a bunch of different types of sugars that they put in, they could see not a ton of activity, but more activity at the surface than at the bottom, and a little bit. So it does appear that sugars were being eaten. But then if we look for the enzymes that are responsible for doing this in the genomes that we're working with, and then we also look for the transcripts, and we plot them on top of this, we can see decent correlations between these Atrobacteria appear to be eating the sugars. And they're the only ones who have this high transcription activity of the enzymes that are eating the sugars. So from this, we infer that the Atrobacteria are the only ones eating the sugars. They're the only ones eating the allantoin. So why don't they just eat everyone else? Why aren't they the only things we see? Like, they're the winners. They're the guys who are the most metabolically active. They've got 10,000 years, 40,000 years. Why not just eat everyone else and win? Well, we look at, so this is a metabolic map of the whole cell that we infer from what we're looking at. The caveat with all this is that we could be wrong. We're inferring things, but it's what we have. You don't have to look at every single pathway, but when they have a box around them, that means that's something that we found in these 20 metabolites. So quite a few of these things were sort of turned on. So they can eat allantoin. They were also, another curious thing that they were doing is that they were making every single amino acid. So they were, which is expensive. Amino acids are expensive to make, but they were de novo synthesizing all of them, which they're the winners. Why are they making this stuff? They can just eat everybody else and not have to make their own. But then the most curious thing of all was when we added up, when we ranked all of the transcripts, these indicators that genes are turned on, the second most transcribed gene in this whole organism you'll never know, is this one. It was an amino acid exporter. Like, they're in an energy-starved situation and they are vomiting out amino acids. They're just sharing them with everybody else, which doesn't seem to make much sense. So we thought about that for a little while. Like, why are they just upregulating this exporter? Why not win? Why not, block everybody else out and win? But there's an advantage. I'm not suggesting that Atrobacteria are altruistic or trying to help their friends. I mean, this is a single-celled organism. That's not what I'm saying. But we tried to look for a reason why they would want to get rid of amino acids, even though they're starving. And it turns out that in E. coli, when the intracellular concentration of amino acids builds up, they will divide. Because it's sort of like pushing a chemical reaction in one direction. Once you get a lot of amino acids built up in a cell, it triggers it to time to make a new cell. So this is a really good metabolizer that makes a lot of amino acids because it needs them. It doesn't want to divide, because dividing is death, because then you make competition for yourself. So the way to win at this game is to be super slow, right? So they have to release them. They have to spit them out just to keep from dividing, to keep them very slow. This is a huge hypothesis. This may all be wrong, but it was the only explanation we could think of that made sense to us. And then this would have the secondary effect of feeding everybody else. So in that way, they get to be sort of keystone species. So also with these other guys, so what about these other guys? Are they alive? Are they doing stuff? Well, another metabolite that we found was trehalose, which is a disaccharide of glucose. And this was present everywhere as well. So this was this really common thing. This one is actually good for keeping proteins from aggregating. So you can kind of think of it as like it does slow a cell down and it keeps it from dividing, but it sort of protects it a little bit. This one, this gene, we were able to find transcribed in more than just Atrobacteria. It was transcribed in quite a few of the different organisms. So this led us with this final sort of big conclusion that maybe trehalose is a universal molecule that allows these organisms to persist in an extremely low energy state for thousands of years. And 45 minutes in, and my daughter is getting antsy. So I'll just quickly tell you that I have another project in Siberia where we found this as well. There, that's the next thing I wanted to tell you. You can hang tight. This is my other student, Katie Sipes, is doing this project in Siberian permafrost with Tatiana Vishnevitskaya here at UT. And she found these same Atrobacteria in permafrost from deep down. So this is now 7 and 14 meters down. And she found that they seem to be producing this trehalose synthase as well. So this is leading us to conclude that maybe this trehalose thing is a general response that everybody can do. So our conclusions are that Atrobacteria might be the keystone species. They may be the most active ones because they can eat things that other things can't. And they make amino acids to feed everybody else. And also this trehalose may be a general molecule to help with the harsh, deep subsurface environments. I want to leave you with the thought that, show it to you first. We didn't just do deep subsurface with our big computers. We also, do me hold you? We also looked at more normal places as well. And what we found is that in lakes and rivers, we also get a lot of these aliens. There's a lot of really weird stuff in lakes and rivers too. Or even wastewater treatment plants, which really surprised us because we thought that this would just kind of be, yeah, like a known thing. No, there's real genetic novelty present in wastewater treatment plants too. So the sort of take-home message I want to leave you with is that the strange new microbes are everywhere, and we don't know what they can do for us. They could find new products, medical products, bioindustry products, or they could do weird things. We just don't know, which I find super, super thrilling. And I'm going to thank the wonderful people who I have funded this because they're beautiful, lovely people. And I'm in Surf. I have my offices on the other side, and I have a two-bridge view, which I'm very excited about. So thank you all for listening.